Hello, my name is Chris McPherson. I'm a principal scientist here at Samsung Biologics, working in the drug substance manufacturing group. I'm going to talk today about how COVID-19 is redefining drug development and manufacturing. As part of my research into this talk, where I came familiar with the term black swan and green swan. Black swans are unexpected, hard to predict events that result in extreme unintended consequences. The coronavirus pandemic is a classic black swan. A green swan as a concept is a positive market and value development once deemed highly unlikely, if not impossible. With the emergence of the COVID-19 pandemic, the pharma and biotech industry has been hit by a black swan. And globally, 335,000 ongoing clinical trials have either been canceled or significantly slowed. Yet there we could also find green swans take off in the cross-pharma collaboration, accelerated drug development, and approvals. Green swan from a CDMO perspective. Technological innovation which enabled speed in timeline, overall time saving by agile execution with predefined platform at each phase and process. Transparent communication and teamwork, expedited batch processing and close communications with clients, and innovative regulatory strategy and support. Successful regulatory submission with strong IP protection. Here's a brief overview of Samsung Biologics manufacturing. Currently, we have 3 plants in operation. Plant 1 has 6 5,000-liter bioreactors. Plant 2 has 10 15,000-liter bioreactors, and then 2 sets of 2 1,000-liter bioreactors. And Plant 3, where I work, has 12 15,000-liter bioreactors for a total of 180,000 liters of capacity. In Plant 3, we have fed batch with N-1 perfusion capability. Currently under construction is Plant 4. Plant 4 has 6 10,000-liter bioreactors, which will be CGMP ready on the fourth quarter of 2022, and the full plant should be expected to be operational for GMP production in 2023. Samsung Biologics is a multi-product facility design with high flexibility and efficiency. The facility was designed to minimize delays to the production changeover, PCO, annual shutdown periods, and maximize production cadence. Slowdown approach eliminates the plants moving away from designed shutdown periods for preventive maintenance and still allow ongoing manufacturing without impacting CGMP compliance through enhanced design and redundant systems. Design production bioreactor turnaround times, 2 production bioreactors for each seed train. Product changeover time is reduced to 0 between different products through the utilization of 2 harvest and 2 downstream trains in Plant 3. Risk-based and phase-appropriate approaches to validation and maintenance to maximize efficiencies. Increased use of hybrid systems, stainless steel and single use to maximize flexibility and internal tech transfers between plants. Optimize timelines for validation and vendor management and technology transfer. Full laboratory services, including process development, process characterization, and testing services are available. The COVID-19 response. Here's a typical technology transfer timeline showing project kickoff, 6 months later an engineering run, followed by 4 months later a clinical run. We start off with producing a gap assessment. From this, we develop the process control strategy and the master batch records. One of the first things that we do as part of the gap assessment is determine the number of capital purchases, as well as the bill of materials for raw materials. The raw materials will have to go through receipt, testing, and release. Small-scale verification runs are done to ensure that we have an effective tech transfer. This typical technology transfer timeline is 6 months or more. The timeline depends on the documentation from the client, the capital equipment delivery, and the raw material receipt, testing, and release. COVID-19 response. Rapid technology transfer and scale-up of COVID-19 projects. The goal was to achieve a tech transfer in as little as 3 months from kickoff to OOF, out of freeze, for the first at-scale run. Our solution at Samsung Biologics was a rapid facility fit assessment, early identification of process gaps, quick sourcing of raw materials, and a risk-based approach to development timeline. It all starts with communication. Communication and collaboration is key to achieving a rapid tech transfer. Rapid communication between the CMO and client. This starts with the facility fit and equipment gap analysis. adjusting the process to fit the facility during development. The COVID-19 treatments were still under development at kickoff. This gave us the opportunity to fit the process to the existing equipment by reducing or eliminating the need for CapEx, long lead time equipment. Client dropship equipment was also available for GMP use. We were able to leverage the client knowledge by both using platform process or a well-defined process to be able to expedite the technology transfer. Open discussions of risk tolerance with the client. Raw materials. Was safety stock actually needed? Or could we run with just enough material for execution of the production runs? Water runs to test automation and equipment. And product quality checked via satellite runs. We need to identify the critical path, early identification, and frequent updates to that critical path. Parallel activities to expedite the program, and identification of bottlenecks. Once we identify these issues, we need an escalation matrix to bring the items to the management and leverage SBL and client relationships with vendors to expedite deliveries. We need to develop contingency plans, a risk mitigation for multiple scenarios. We also need to work with communication collaboration, not just between the CMO and the client, but between the CMO, client, and vendors. Early identification of process gaps and long lead time raw materials, such as medium components, chromatography resins, and filters for viral filtration, ultrafiltration, diafiltration, and final fill. Adaptation of the process to meet raw material requirements, such as chromatography resins. If an anion exchange resin was needed, and the one that was initially chosen by the client was no longer available at commercial scale, was another ion exchange resin available that would suit a similar need. Reallocation of purchase orders to reduce lead time. Ordering medias and components by the client, and then switching those purchase orders over to the CMO allowed us to reduce the lead time and get our raw materials on site faster. And risk mitigation plans if raw material shortage is experienced. Were alternate vendors or materials available, such as filters for viral filtration, UFDF, or the final fill. And using enhanced communication technology, virtual meetings. From the very start, clients were not able to come on site due to the global pandemic. So we had to have clients conferences right from the initial contact, including data sharing and client access to our quality and compliance information systems for approval of documents. When the clients performed audits, whether it was a due diligence or an ongoing client inspection, we did virtual tours of the manufacturing area using gimbal-mounted high-definition cameras and omnidirectional microphones. This enabled two-way communication with the subject matter experts in the manufacturing area, where we could actually zoom in and out on various pieces of equipment or processes as they were ongoing to be able to show clients what was happening on the manufacturing floor. We also used virtual communication with the vendor meetings. We could do a three-way conference between the vendor, client, and CMO for data sharing. We're also able to do inspections with regulatory agencies through a virtual inspection, where we did virtual tours of the manufacturing area and had communication directly between the subject matter experts with the inspectors from the regulatory agencies. Risk to benefit with accelerating timelines. One of the ways to accelerate timelines dramatically is to remove engineering runs. During a standard process, an engineering run provides value. It allows for the testing of process parameters at scale, check of the automation and equipment controls, and hands-on training for operators for the new process. An accelerated timeline, when we have a well-characterized process or platform, little adaptation required, as well as well-understood risks with mitigation strategies in place, we are able to remove the engineering run. Removing the engineering run can accelerate timelines by removing 1.5 months of engineering run execution, QC data analysis for the engineering run, as well as updating the documentation based upon those results. Overall, this can eliminate up to 4 months in the timeline. It can only be done when we have that well-characterized process or platform, little to no adaptation, and well-understood risks. I want to take a little bit of time to go over 4 case studies that we experienced during our rapid technology transfer. And the first case study was about raw material availability, the virus filter supply issue. The background and problem was identified due to the global shortage in virus filters. Flexibility was required in which filters were chosen. We had discussion with multiple vendors to secure the needed supply, not for just the initial at-scale runs, but also for the upcoming campaigns. Collaboration with the clients to identify alternate options, the development history, clinical study, and manufacturing history was taken into consideration. And contingency plans were developed collaboratively between Samsung Biologics and the client. The result was a change to the virus filter during development, potential change to the virus filters for future campaigns. In case study number 2, raw materials testing and release, the master cell bank and working cell bank availability during the technology transfer was an issue. Due to the COVID-19 quarantine, master cell bank and working cell bank were under development during the technology transfer, and full testing was not available. We revised the requirements for shipment under quarantine and GMP release separately. Shipping was done under reduced testing. Quarantine release upon receipt of the test results, and a full receipt upon receipt of the C of A. This allowed for expedited shipments while ensuring quality, allowing us to get the master cell bank and later the working cell bank here on site for use both in the small scale runs as well as in the at scale runs. The result was the testing and shipment procedures revised and the timeline was accelerated. In case study number 3, process documentation. The process documentation was not available at the start of the tech transfer. The process was under development at the client site, and limited historical data and process documentation was available. Frequent updates were being carried out as studies were completed. We solved this by doing a rolling document update. Ad hoc meetings were called with the client as information became available. Documents were revised sequentially for each unit operation, and we reduced the document review cycle times for approval. We established a joint communication committee together with a structured communication plan. And as a result, with collaborative efforts filling in the gaps, data sharing in real time, and reduced documentation cycles, we were able to achieve a rapid technology transfer. In case study number 4, about equipment availability, we had a shortage in seed train bioreactors. The lack of seed train bioreactors to use in the client process at the time, we could source from a local vendor was possible, but we wanted to ensure comparability between the site client seed train bioreactor and the bioreactors provided by the local vendor. For the initial scale run, internal transfer from another SBL plant, which was located on the same campus, was done. However, for the continuing campaign, those bioreactors were going to be needed back in the original plant. So for the production campaign, a limited number of equipment was drop shipped from the client. We then secured the rest of the additional equipment from a local vendor. We overcame the importation of requirements through close coordination between the client and SBL. Then our engineering team was able to ensure technical alignment between the drop-shipped seed train bioreactors from the client and those secured from the local vendor through a fast execution of IQ, OQ, PQ. The result, through a complex series of internal and external equipment transfers enabled by a close collaboration between Samsung Biologics clients and a local vendor, and leveraging Samsung Biologics plants at one site, equipment availability issue was resolved and the timeline was kept on track. I want to thank you for taking the time to listen to this talk, and I appreciate any comments or questions that you have. Thank you.